first slide, I just put my medical mycology kasi marami namang branches din of mycology. So you see the different images there. Yan yung mga makikita nyo later on. If we'll be able to have even just yung sa before your internship or during the internship na walang face-to-face when we will be meeting you siguro sa school or hindi ko pa alam. It will be dependent on the outcome of the meeting this afternoon. Okay, so first, before I start, I would like to give again credits to Ma'am Ethel and Sir Reynald, to whom this PowerPoint was based. Sila na rin ang most of this came from them actually. So, nag-update na lang ako, okay? So, looking at the pictures, so alam ko, if you look at an introduction to mycology kasi is that we will now be talking about the fungi or the kingdom. Fungi, maingay yung aking aso, or what is also called Mycetea, which is a large and filled with forms of great variety of complexity. But for practical purposes, we can divide them into two groups. So that we have the, patigilin muna natin yung aso. Okay. We have the macroscopic fungi. Ano yung mga macroscopic fungi natin? So you could see a picture at the lower left. This would be our mushrooms, yung mga puffballs, and also yung gill fungi. And the microscopic, so that if you notice, ulit sa slide natin, we have the bread. Underneath is yung picture. Ano ba yung tumutubo na yun? Ano ba ang mold na tumutubo? Ay natawag natin amag. So that is it a living cell? So you see there the illustration on what actually is growing dun sa ating bread. Then, it's also true, nakikita nyo lagi yan sa ating mga kamatis and strawberries. Okay? So that with this, we can already come up with the definition of mycology. Ano ba si mycology? So that we say now that, you'll get it in your next slide later on, na ang mycology is the study of fungi. What about the fungi? This will include their taxonomy, nakasama dun sa prior, activating knowledge, yung APK ninyo, activating prior knowledge. So it will include also the taxonomy. So again, it was there, either there you have your botanical taxonomy or according to the type of infection that they cause, and also their environmental impact, genetic, and biochemical properties. So lahat yan kasama sa mycology. Now, lower do sa slide, you should also see some mycosis, diba? What is mycosis? Sabi natin fungal infection. Any infection that is caused by a fungi. So that, with that, we can now also come up with the definition. Kita nyo yung mga, the most common of which is yung nasa lower right. Yung tinatawag natin na, alam nyo kung ano yan? Joshua? Asa na si Joshua? Ano yung image sa lower right?
Ma'am, iniisip ko nga po kasi may ganyan po yung pinsan ko eh.
Very common na yan sa atin.
Yes, ma'am, nakalimutan ko po yung tawag dyan.
Ano sa Ilocano, sa iba na?
When, ma'am, may... Parang, ang sabi nila, ma'am, bungang araw, pero may iba pa pong term dyan, ma'am.
Hindi, ang layo niya sa bungang.
Hindi, ma'am, ano kasi tawag dito?
Bungang araw. Wait lang, mga ano. Wait lang, mag-close lang ako ng window. Maingay yung hindi bungang araw yan, George.
Ayun, ma'am, kamanaw.
Kamanaw. O ano pa yung ibang word ng kamanaw?
Ano pa kasi?
Pag nakita nyo merong puti-puti, parang anong tawag doon? Very common yan na superficial cutaneous mycosis. Narinig ko pa rin yung aso. An-an. Diba? So those are fungal infections. Now, you will see later on, you will learn later on din na meron tayong classification of fungi na according to the site or the type of infection. So that we come here now into the definition. Sasabi natin kung ano na ang mycology. And then, of course, we have medical mycology, which is the science devoted to the study of fungi and their relationship to human disease. It is also defined as a scientific discipline which encompasses single-celled yeast and filamentous molds as agents of disease. So now our fungal agents include the true pathogens and opportunistic pathogens. So review ulit ng bacteriology ninyo. Pag sinabi natin true pathogens, ano yung mga yan? True pathogens. What are true pathogens? Ay, bakit? Okay. True pathogens. Alex. Yes, Alex. Again, you can use your chat box. Serena. True pathogens and opportunistic pathogens. Okay, so thank you, Serena. Okay, sa true pathogens, kahit healthy ka, kahit immunocompetent ka, pwede ka pa rin magkaroon ng infection. They are still able to cause the infections. In contrast to the opportunistic, kailangan immunocompromised ka. Okay? So that the terminologies here, this is our activity 1. So some of them have been mentioned already. Just get a piece of paper. Meron naman siguro kayong notebook o kahit anong papel dyan sa tabi ninyo. Now, in your paper, write a word. One word lang ha. One word that can define or that can give you any insight into the following terminology. So you have there your mycosis. 1 minute lang. 1 minute. One word lang eh. Mycosis, demacious fungi, dimorphic fungi, imperfect fungi, perfect fungi, polymorphic fungi, mold and yeast. Go. Nakatingin ako sa timer. 1 minute. Tabi niyo yung papel niyo ah. Thirty seconds. Okay, time is up. We'll start. Tingnan natin. Sinong una? Can I have Ira? Ira, may mic ka? Brandon. So si Brandon.
Morning. Ay, good afternoon, ma'am.
Good afternoon, Brandon. One word for mycosis.
Infection po.
Okay, very good. Demacious fungi.
Ako rin po ba, ma'am, or iba na?
Ha?
Ako pa rin po ba, or iba ng
Ikaw pa din. Lahat yan, bibigyan mo ako ng isang word. Demacious fungi.
Wala po akong maalala. Hindi ko
Okay, dimorphic.
Ma'am, both yeast and mold
One
yeast.
word lang eh, one word. One word lang.
Wala. Hindi ko mo ganun.
Imperfect fungi.
Asexual.
Okay, asexual. Perfect fungi.
Sexual, ma'am.
Polymorphic fungi.
Wala po, ma'am. Hindi ko maganda.
Mold.
Fuzzy.
Ha?
Fuzzy.
Okay. Yeast.
Tawag dito. Lat. Ay, parang, tawag dito. Wala. Hindi ko maganyan. No? Hindi ko
Okay.
in one word.
Okay. Thank you, Brandon. Let's get into last na pumasok ngayon. Asan si Alfred? Alfred. Ano si Alfred?
Opo, ma'am.
Okay. One word. One word, Alfred, for the different
Po?
terminologies. Isang word just to define na maano natin kung ano yung word na yan.
Anong ano po?
Yung nasa screen mo.
Saan po dyan, ma'am?
Lahat. Mycosis, demacious fungi, dimorphic,
Ay.
imperfect, perfect, poly, mold, yeast. One word for mycosis.
Infection po sa
Okay,
mycosis.
demacious fungi.
Mycology fungi.
Yeah.
Pigments po.
Okay, very good.
Pale.
Okay. Dimorphic.
Dimorphic. Ano po, ma'am?
Ano?
Dimorphic.
From the prefix di.
Yeast.
Ha? Imperfect fungi.
Wait lang. Ayan. Wait lang. Sa ano po, sa imperfect fungi po yan, ano?
One word. Asexual
po.
Okay.
Tapos
Perfect.
sa perfect po, ma'am, is sexual.
Poly.
Tapos po sa polymorphic fungi is same culture.
From the word, prefix na naman, poly. Okay, mold.
Sa mold po is woolly po.
Okay.
Woolly.
And
Tapos
your
sa yeast.
Yeast?
Ano po, ma'am? Probiotic.
Ha? Okay, thank you. Thank you, Alfred. Let's get into one more. Harley. Asan ka si Harley? Harley?
Hello, ma'am.
Good afternoon, Harley.
Good
Kumusta?
afternoon, ma'am. Okay lang, ma'am.
Okay, your chance. One word for the different terminologies.
Ma'am, yung mycosis, infection din po yung nasulat ko, ma'am.
Okay.
Tapos, hemataceous is dark.
Yes, very good.
Dimorphic, both, ma'am, both. Kasi it
Okay.
involves two phases. And
Yes.
perfect,
Okay.
asexual. Perfect, sexual, ma'am.
Oh,
Polymorphic, many. Madami.
yes. Madami.
Mold,
And-
ma'am, is fuzzy. Then yeast is
Okay.
creamy.
Okay. Okay, very good. So, with that, kasi pag ganyan, hindi nyo kailangan talaga mag-memorize, di ba, ng mycosis is an infection caused by, that's an infection caused by a fungi. So, ako kasi kanina, naisip ko lang yan while going over the PowerPoint. Sabi ko, gawa nga ako ng activity din dito. So, that demacious, sabi nga ni Alfred, di ba, pigment. So, that demacious, ang nilagay ko sa akin, may kulay or just kulay. Kasi yan yung mga merong pigments because of the presence of of melanin. And then dimorphic from the word di, sa akin dalawa. You have two phases. You have the mold and the yeast phase. And then imperfect, sabi ko imperfect kulang. Si perfect, perfecto, di kompleto. Imperfect, they lack the sexual reproduction. Si perfect meron. Polymorphic, parang sinabi din ni ni Harley, madami. And then the mold for me is, ang dami yung pwedeng gawin sa mold and sa yeast. Si mold, may hyphae. Si yeast, walang hyphae. Si mold, multicellular. Si yeast, unicellular. So, yung mga yun, that's how you can get into the different terminologies. So, a while back din kasi kanina, titingin ako, meron na naman ako nakita sa na meron silang mga terminologies specifically for mycology lang. So, you can get into that also when you have an extra time to go over those things. Okay? So, moving on, we have now, let's get into the epidemiology. You see here that humans are relatively resistant to infections caused by fungi except for the dimorphic fungi. So ito lang yung mga pwede na tayong magkaroon ng infection. Then what are the different predisposing factors for the host to get the infection. So one of which is, sabi nga natin kanina, they're considered to be opportunistic. So pag immunocompromised ang host, pwede ka magkaroon ng mycosis. So ano nangyayari? Bakit nagiging immunocompromised si host? Is because also of immunosuppressive agents or meron silang underlying diseases o yung tinatawag natin ngayon na meron silang comorbidities. Then, pwede rin sila nagkaroon ng complex surgical procedures which in consequently na compromise din ang immune system. And of course, when nagkaroon ka ng medyo prolonged antibacterial therapy. Okay? So those are the major predisposing factors as to acquiring or yung magiging prone ka or mai-infect ka nitong mga fungi. Now, generally, they are not communicable so that humans acquire mycosis through first inhalation spores. So merong picture doon, you breathe in the fungal spore. Okay? And then it gets into the respiratory system and then yun na nga, kung hindi ka immunocompetent, pwede kang mag-develop ng infection. It can also be through direct contact with the spore. As shown again, tinawakan mo si cat, may fungi pala dun sa kanyang fur o sa kanyang skin, so may direct contact with the spore. And then inoculation by trauma into the skin. Classic examples is porotrichosis na sa mga kaya tinawag na rose gardener's disease din siya kasi pag natusok ka nung thorn nung rose na nagkataon meron dong spore nung fungi, so pwede ka rin magka-develop ng sporotrichosis. So those are your modes of transmission as to fungal infections. Now, as to the general characteristics ng ating fungi, okay, so we say that they are eukaryotic. They're considered also are classified as talophytes. Why? Because they have a true nuclei and they are also heterotrophic. When we say heterotrophic, anong ibig sabihin na ulit? If you go back to your bacteriology where you had your autotrophs, you had your lithotropes, you had your chemotropes, chemoheterotrophs, so that when we say heterotrophic, ano na ulit? Anyone? Heterotrophic. Para hindi nyo makalimutan yung mga iyong terminologies. From the prefix again, hetero is kanila kinukuha lahat ang nutrients nila. They're considered heterotrophic because they acquire nutrients from a wide variety of organic materials which we call substrates. Kaya nga tinatawag din sila kuminsang saprobes. Bakit naman sila saprophytic? Why do we call them saprophytic in nature? Or bakit natin sila tinatawag na saprobes? It's because they also rely on saan? Saprophytic or saprobes. Sa mga substrates from dead or remnants ng dead plants and animals. Tinatawag din silang parasites so that if you get to some books, they're also considered as parasite. Bakit naman sila considered as parasites? Kasi they can live on the bodies of living animals or plants. So nagdidepende naman sila dun sa living animals or plants for their nutrition para sila able to grow and then also multiply. Now they have chitin in their cell wall and ergosterol in their cell membrane. As you all know that they lack chlorophyll, kaya nga they cannot produce their own food. So there is also a lack of susceptibility to antibacterial antibiotics. For the obvious reason, bakit sila hindi sila susceptible to antibacterial? We have different modes of action sa mga ang mode of action ng antibacterial drugs, di ba? So these are fungi. So we also have our antifungal agents, okay? So another is they're also obligate aerobes. Almost all molds are aerobic, but most are facultative and aerobes. Now, they can grow on a very low moisture content. Basta may moisture, carry na yan ng mga fungi. Pwede na silang mabuhay. And they require less nitrogen than bacteria, and they're often capable of metabolizing complex carbohydrates such as lignin. Now, activity 2. Keep your papers, ha. Kukunin ko mamaya after ng ating discussion. You're going to send it sa PM or sa GC. Check ko yan. Activity 2. You have learned about your fungi. Now, let's try to differentiate or compare your fungi and bacteria. And before I forget, sorry ha, I will interrupt. Doon sa activity nyo, whenever you are asked to differentiate, remember the first question was differentiate sexual from asexual. Hindi nyo kailangan i-define o i-describe ano ang asexual. You get into the differentiating factor. Usually yan, pwede kayong magganito din. Gumawa ka ng ano ba yung mga factors ma-differentiate ko si sexual from asexual. Avoid, describing the process. But rather, dwell on the points that you can differentiate one from the other. Okay? So, ito ngayon, let's try to differentiate your fungi from the bacteria. As to the presence of the nucleus, you have the mitochondria, yung cell wall nila, ano ba meron? Sa cell membrane, provide ko ang sterols absent, sabi ko sa bacteria, and the rest. So there are 11 there. Okay, go. 1 minute. 1 minute lang, ha? Fifteen seconds. Okay. Says, can you present your output? Andiyan si Says. Bayukan. Princess, are you there? Wala si Princess? Says. Wala si Says. Charlene. Charlene?
Yes, ma'am.
Okay, ano answers mo first sa nucleus?
Sa fungi po, ma'am, has nuclear membrane. Tapos
Okay.
sa bacteria po, no membrane.
Okay, how about for the mitochondria?
Ano po, meron siyang mitochondria, tapos sa bacteria po, walang mitochondria.
Okay, very good. How about their cell wall?
Sa fungi po, it is made up of chitin po yung cell wall, tapos sa bacteria is peptidoglycan po.
Okay, thank you, Charlene. Juna V, continue. Where's Juna V?
Hello, ma'am.
Yes, good afternoon. Continue. So, nasa cell membrane tayo.
Yes, ma'am. Tapos sa dimorphism po, kapag fungi, meron tayong molds and yeast forms po. Kapag bacteria, wala po.
Okay.
And sa sedimentation coefficient po, when it comes to bacteria, it's 70.
70S. Ano ulit ang
70.
ating 80S and ang ating 70S? Ano yung mga yan? Ano si 70S at saka si 80S? Ano yung mga yan? Kaya tinatawag natin na sedimentation coefficient. Ano ang responsible dyan sa ating sedimentation coefficient? You had that also in your bacteriology, di ba? Doon sa ating prokaryotes. What is your 70S and your 80S? These are your ribosomal subunits. Okay, ano ang role ng mga yan, ng mga ribosomal subunits? So that you have the 80 ribosomal subunits for your eukaryotes. Then you have your 70S ribosomal subunits for your prokaryotes. Okay, so yan kasi, yan yung sa ating, ang ginagawa nila. Ito yung mga nagpe-play ng role sa catalysis na important sa biological processes. Okay? So that, yun yung sedimentation coefficient, di ba yan yung measure ng rate at which yung sa mga proteins, yung mga molecules suspended in colloidal solution sediments in a ultracentrifuge para yan dun sa ano na yun sa ating mga sa function din ng ating ribosome. How about for the cultivation?
Less than 6 po, ma'am, yung
So
pH.
ano yung environment pag less than 6?
Alkaline? Ay, acid.
Nalito na si Juna.
Acidic.
Hindi ka nag
Acidic po. Sorry po.
Acidic.
Sorry
Okay?
po. Yes,
So,
ma'am.
nucleus, they have a defined nuclear membrane. Si bacteria, wala. Si mitochondria, present kay fungi, kay bacteria, absent. Si cell wall ni fungi, made up of chitins. You have also there your glucans and your mannans. Your bacteria, you have your peptidoglycan. Cell membrane, anong meron sa cell membrane? You have your sterols or what we said earlier dun sa general characteristics, the ergosterol. And of course, since banggit kay bacteria, meron siyang respiratory enzymes. Si fungi, wala siyang respiratory enzymes. Okay? Dimorphism, of course, present sa fungi, wala sa bacteria. Sedimentation, 80S, 970S sa bacteria. Sabi nga natin kanina, eukaryotes, ribosomal subunit 80S, prokaryotes, ribosomal subunit 70S. Cultivation, acidic, kaya less than 6, pH of 6. Your bacteria, 7.2 to 7.6. Okay, thank you again. So we get into the morphologic form. Sabi nyo kanina dimorphism or the types of cells na meron ang fungi. Di ba sabi natin kanina when we divide them into the macroscopic and the microscopic fungi. So that in the microscopic fungi, you have the molds and the yeast. So ang mga yeast natin, sabi natin, di ba, they're unicellular, that's a single vegetative cells. They're usually spherical or ellipsoidal. They grow from 35 degrees centigrade to 37 degrees centigrade. Now, ano ang colony characteristic nila? That usually they are mistaken for also a bacterial colony kasi parang pareho lang ang itsura nila. So that the colony is moist, creamy, opaque, or pasty, resembling bacterial colony without aerial hyphae. Okay? You take note of that. Pag yeast, walang aerial hyphae. The identification of these yeast, it is based primarily on biochemical testing. So few of these is divided by binary fission, but most reproduce asexually by budding. So you'll learn more about budding sa succeeding slides. So for the molds naman, so if you will be asked again to compare your molds and your yeast, marami kayong magagamit. Okay, unicellular si isa, multicellular si isa. Merong hyphae si isa wala. Okay, now after their colony, you look at the petri dishes with molds growing on them. So makita nyo yung fluffy, cottony, woolly, or powdery na characteristic nila. Obvious naman yan, except pag yun nga kay yeast, hindi natin ma-differentiate kaagad kay bacterial colony. Ang kanilang growth temperature is at 25 degrees centigrade. Now, as to their identification, you get into their growth rate. Growth rate is since certain fungi or most of them, meron silang specific number of days bago mo sila makitang mag-grow dun sa media. So that with the mold, this is very important. Kaya pag, again, we can try this one. I can prepare the SDA. Yung mga nasa Tugigaraw, pwede ko kayong bigyan and then you try. Kaya lang, I'll learn later on still of the biosafety. Para lang makita natin. Or we can do this later na at the end of the semester. Pag medyo maluwag na siguro ang ating mga regulations sa ating city. Okay, growth rate. So once may nag-grow doon sa inyong media, you start observing it kasi sure naman na pag hindi tayo talaga nag-inoculate ng fungi doon at nag-expose ka lang sa iyong surrounding, lalo na sa medyo moist area, meron mag-grow dyan at hindi lang iisa. Okay, so aside from their growth rate, we can use also their macroscopic and microscopic appearance. So another example there is you see now your Rhizopus colony. Diba? Sorry. Rhizopus colony, more of hindi masyadong mataas. Kung ganito lang kasi we're looking at it top view. So sabihin natin powder yan. Now you get a colony of that and put it onto your slide, apply your stain, and then you check its microscopic appearance. Makikita nyo na ngayon yung fungal elements or the different fungal structures that are specific to Rhizopus species. Okay? Sorry. So you will learn about the different structures as we go along. Now, dimorphism versus polymorphism. So, ano na lang ito mga bullet-bullet kasi these were all listed from Mayhon. Diba? Sabi natin, I already mentioned to you that majority or yung major book na gagamitin ko for all our lessons will be on Mayhon. Not unless you want to add more, then pwede lang you can go read your Baileys or whatever micro book that you have. Sabi ko nga, you do not need to concentrate only on what is given to you. Magbasa-basa din tayo ng iba. Okay? So, dimorphism, na ano nyo na kanina yan? Nasabi na yan ni na Harley, nasabi na ni Brandon, at saka ni Alfred. So, dimorphism, dalawa. There are two states wherein fungi can exhibit, pwedeng yeast or tissue state, pwede yung mold phase. So, saan sila nagkakaiba again? So, yung sa kanilang temperature na mag-grow sila. So, your yeast or tissue state, kaya tinawag na tissue state, ano ba yung 37 degrees centigrade natin? Diba body temp yan? So, that sa labas niyan, pag nagkukos sila ng infection, they're in the mold phase or the mold state. Pag nakapasok na sa katawan niya, na-infect na tayo din, ano na siya into the tissue state or the yeast phase. That is why you see here that the mold phase, they grow at room temperature 22 to 25 degrees centigrade in ambient air. So, yun yung sinasabi sa akin kanina ni Jean. Ma'am, room temp po ang linagay ko. So, I'll go over your quizzes, your pre-test mamayang gabi na kasi may klase din ako right after your class. Then, we have here the thermally dimorphic fungi, mga examples siya, which you will meet then later din sa ating mycology. And the polymorphic fungi, sabi nyo nga kanina, marami. So, marami in a sense that both the yeast and the mold forms will be seen in the same culture. So, paano yun? Example natin is yung Exophialis species. Di ba sabi natin, they can first stay in a mold phase and then pag nakapasok, magiging yeast. With the Exophiala kasi what happens is mag-ano muna siya, yung first phase niya is yung sa yeast phase. Habang nagmamature siya, doon sa plate niya na kunyari, kinoculture natin siya. Habang nagmamature sa Exophiala, doon siya mag-exhibit ng kanyang mold phase. So that is what we call, kasi theoretically, mag-exhibit ka lang muna ng isang phase at a certain condition. Di ba? Sabi natin, mold would grow at 22 to 25 and then the yeast would grow at 37. Pero kung Exophiala, at the same condition, pwedeng nag-grow muna siya as yeast and then, habang nagmamature siya, naging mold phase siya. Yun yung ating polymorphic fungi. Now, for the general morphologic features na tayo ng molds. Now, we have, of course, the hyphae or the hypha, depends on how you want to pronounce it, which is a tubular branching filaments of fungal cells. It is the mold form of growth. So, yung nakikita ninyong cottony na sinasabi natin na colony morphology ng mold is because of this hyphae. It is considered the microscopic unit of fungi. So, pag tinanong ko kayo later on, if you have your post-test and then sa microscopic unit of fungi, huwag kayong sasagot ng mycelium. It is the hypha or the hyphae. Okay? So, ang hyphae natin, pwede siyang septate o pwedeng acetate. When we're talking of septations, it is the presence or the absence of cross walls or what we call now the septum or the septa. So, ang example ko dyan lagi is to look at your fingers. So, nakikita niyo yung division nun. So, that is how a septate hyphae looks like. Merong division. Si aseptate, wala. Although, dito kasi sa aseptate, kung minsan, medyo magkakalayo yung cross walls nila. So, meron tayong wala talaga, meron naman tayong magkakalayo, that would be your sparsely septate, and then, o kaya yung sinusitic hyphae. Okay? So, ito yung examples natin dyan. So, you have a Phaeoacrimonium species displaying septate hyphae. Obviously, you can see there the cross wall. Okay, may division. Compared to your Mucorales hyphae, which appears sparsely septate. I've been trying to really look into work bakit sparsely kasi dapat magkakalayo lang. Pero meron pa rin siyang cross wall. So dito, makita mo yung nasa 9 o'clock banda. Meron dyan, merong septation na hyphae. Pero malayo kasi yung septation niya. So that's sparsely septate. In contrast, pag sinabi natin aseptate, na kahit tinignan mo yung buong hyphae, wala talaga siyang cross wall. Okay? And then, aside from the presence or the absence of a septum or a septa or a cross wall, meron pa rin tayong differentiating appearance or tawag natin dyan, itsura ng ating hyphae. So, meron tayong tinatawag na antler, meron tayong tinatawag na racket, a spiral. All those words speak for themselves. Pag sinabi mong antler, yung kasama ng parang si reindeer, yung kanyang, hindi ko alam kung pwedeng tawagin sungay yun. So, that is your antler. So, pag nakakita tayo ng ganun, sa microscope and as a slide on a prepared temporary mount, that could be your Trichophyton schwenleni. Remember that we said earlier, for molds, they can always be identified by, first, sabi natin, their growth rate. Second is their macroscopic appearance. And then microscopic. So, nung nakita mo na siya, yung mold form of growth niya, either colony, fuzzy, or we said powdery, and then gumawa ka ng iyong temporary mount, gumawa ka ng slide mo, tinignan mo, meron siyang antler hyphae. Sabi mo na, this is Trichophyton schwenleni. Kasi like for your racket hyphae, from the word racket, mukha siyang tennis racket. Meron siyang, nag-enlarge siya doon sa dulo. Parang itsurang tennis racket talaga. Of which this is exhibited also by your Epidermophyton flocosum. Sorry for the C there. It's Epidermophyton flocosum. And another one is your spiral hyphae. So, spiral, alam nyo yan, coiled, showing corkscrew-like turns, which is also exhibited by our Trichophyton mentagrophytes. So, ito yung mga pictures lang nila. So that, ayan yung illustration sa taas. So, you have your spiral hyphae, pectinate body, fabric chandelier, or the antler hyphae. Then we have our nodular organ and the racket hyphae. Yan naman, pag tinignan natin sa slides, ito yung kanilang itsura. Okay, you have there the spiral for mentagrophytes, you have the antler for schwenleni, and you have the racket hyphae for our Epidermophyton flocosum. Now, let's get into the hyaline versus the phaeoid. Sabi natin kanina, phaeoid, di ba sabi ulit ni na Harley, Brandon, and Alfred. Sa akin kanina sabi ko, kulay. Sabi ni, sino nagsabi ng pigmentation? Was it Brandon or was it Alfred? Pigmentation. Okay. So, when we say hyaline, nagpato na ba? Naghisto na kayo? Naghisto na kayo? Di ba naghisto ba kayo ng second year? Junavi? Huwag niyo akong tulugan. Alam ko, pag ganitong oras, nakakaantok talaga. 1 to 3. 1:30 to 3:30. So, when we say hyaline,
Yes, ma'am.
pag hyaline Tam, hindi tulog si Tam ngayon. O Tam, hyaline versus phaeoid. Exercise muna ako, Tam. Tam.
Ma'am, pag hyaline po lightly pigmented, kapag phaeoid naman darkly pigmented dahil sa melanin.
Okay. So, yun na sinabi niya. Darkly pigmented. So, may pigmentation. Bakit may pigmentation? Gawa ng melanin. Now, with this, nandito sa slide yung ating Mason Fontana stain, which is used primarily or specifically to determine hyphal pigmentation in tissues. So that pag yan ay phaeoid, yung kanyang hyphae, we're still sa hyphae ha, yung stalk lang yan, magkukulay siya ng brown. Pero pag siya ay hyaline, siya ay magkukulay ng pink or pink to red. Okay? So, yung example natin dyan, so nakikita nyo yung example natin sa phaeoid, yung kanyang dark pigmentation is, again, due to the presence of the melanin sa kanyang cell wall. Now, let's get into the general morphology. Still pala, still on the general features of our molds. So, di ba sabi natin kanina si hyphae is the microscopic unit of mold. And a group of hyphae now, like the group, parang yung fingers nyo, bawat isa dyan, hyphae. Pinagsama-sama nyo lahat yan, yan na ngayon ang ating mycelium. Yun ang imagination. Yun ang ating parang tawag doon, correlation siguro. So that we define mycelium as a mass of intertwining structure composed of hyphae. And these accumulate during active growth. Now, yan, imagine mo yung halaman o yung mismong mushroom pag tumutubo yan. So that merong pataas, merong pababa. Both of those are mycelium. Diba? So that yung nasa taas, that is your aerial mycelia, and it is also called as your reproductive portion or reproductive mycelia for the main reason na nandito yung ating mga fruiting bodies. Na yung fruiting bodies na yun, parang reproductive organ na rin nila or means of reproduction, yun yung mga nagproproduce nga ng conidia or spores. Taken, kasi yan, pwedeng taken interchangeably kasi parang pareho lang naman. Depende lang kasi either product of sexual or asexual reproduction. And yung aerial natin, kaya nga aerial, they extend above. Yun yung tumutubo pataas. Okay? Yung ating vegetative portion or the vegetative mycelia, yun yung parang roots. Consider natin roots. Siya yung nag-a-anchor ng colony natin. Siya na rin yung nag-absorb ng water and nutrients. So it gets into the culture media. Okay, yun ang ating vegetative mycelia. Now, let's get into reproduction. Kasama ito dun sa inyong activating prior knowledge. Paki-explain asexual and sexual reproduction. Wala pa rin si Cez. Ira. Diyan ba si Ira? Wala si Ira. Judrit. Andiyan si Judrit? Judrit? Wala din? Si Maridane na lang. Maridane. Andiyan si Maridane.
Yes, ma'am.
Maridane. Explain sexual and asexual reproduction in your own words.
Ma'am, asexual reproduction is considered imperfect reproduction po. Tapos, dito po yung no need na po ata yung meiosis, ganun, mitosis lang po pwede na. Tapos, meron po siyang 4 types po ata, ma'am.
Okay, sige, go.
Tapos, yung for sexual reproduction po, ito po yung perfect reproduction kasi meron po yung parang dalawang panggagalingan nung cells to create fungi or to create colony po. Tapos meron din po siyang 4 na types po.
Okay.
Yun po.
Okay, very good. So na-explain naman clearly so that we say kasi that most fungi can propagate by the simple outward growth of existing hyphae. Yun yung sa ating asexual from the hyphae, di ba? Either through fragmentation, sa example niya, nag-drawing kayo ng arthrochonidia. So by fragmentation, kasi pag naghiwa-hiwala yun, bawat portion nun magde-develop ulit ng panibagong fungi. Okay? Tapos, doon ulit yung magde-develop ng bagong colony. So that the primary reproductive mode of fungi involves the production of various types of spores. Kaya mga fungal spores na yan, hindi lang sila responsible for multiplication, but also for, parang balik kayo ng inyong bacteriology, also, although it should not be confused with the endospore sa bacteriology, ha? Itong mga spores din dito sa ating mycology, they are also produced for survival din. Kaya dun sa isang example ninyo, di ba, meron kayong chlamydospore. Pag hindi maganda ang environment, magdo-dormant muna siya. Pag favorable, mag-germinate ulit siya. Okay? So, through that, magproduce din yung mga spores na yan, especially with sexual spores, ng magkakaroon ng genetic variation kasi hindi in contrast sa asexual na you'll be producing the same kasi nag-bud off lang siya doon sa mother cell. The same. Although pag sexual kasi, kaya nga sinasabi natin two parent cell, parang ganun ba, dalawang cell na magkaiba. So, pwedeng magkaroon ng variation. Hindi sila magkamukha ngayon, no? Parent cell. Okay? So, we have different spores nga that are produced by means of this sexual or asexual reproduction. So that here we have the sexual spore. So, yung fungi natin, they contain, what do you call this? They contain a marked diversity of spores that they are largely classified and identified also by their spores and spore-forming structures. So that the most general subdivision is based already on the way the spore arises. Okay, so that sexual spores now you have your ascospore, you have your basidiospore, then you have your zygospore, and then you have your oospore. So madali na lang talaga silang tandaan kasi pag sinabi mo contained in a sac-like structure called ascus, alam mo na kaagad kung ano yan from the word ascus. So following what happens, so you know the process na ina-undergo niya bago siya maka-form within an ascus. Ang ating so this is just an example. Like if you have a cleistotium, what is a cleistotium? This is a sexual fruiting body din. Pag nag-rupture siya, so magre-release siya ng numerous ascospores. Okay? What will happen to these ascospores? Kasi nga, these spores of fungi, did you ever wonder, bakit kaya merong tumubo dito na mushroom? Like, for example, I was thinking of the bread. Kasi kung babalik na naman tayo ng theory, spontaneous generation and all, sabi ko nga, paano nga kahit nagka-mold yun, hindi mo naman binubuksan yung tinapay. Then I came into, sabi ko, di ba may yeast ang tinapay? Kaso yeast, bakit lumabas si mold? Yung mga ganun lang, naiisip mo pag ganyan, nakakakita ka ng molds na nag-grow sa tinapay o dun sa ating mga vegetable or fruits. Now, yan mga spores kasi they're light. So that pag humangin, na ihahangin sila anywhere, bumagsak sila, and if the environment is favorable, mag-germinate, and then mag-grow ulit, and then develop again to this conidia or spore or to the fungi. Okay? So, the next is our basidiospore. Sabi ko nga kanina, kung ascus is to ascospore, now you have the club-shaped basidium, which is now also inside that basidium is your basidiospore. So that following meiosis, 4 meiospores usually form on the surface of a basidium. Now, before I forget again, when you are asked to illustrate, I don't know if I did mention this to you before sa bakte natin, kaso hindi tayo nag-drawing masyado, make sure that you always label your drawing. Whatever relevant, hindi naman basta-basta na maglagay ka na, hindi naman hinihingi. Whatever that is relevant to what you're trying to illustrate, please put labels on it too. Okay? So, ito naman yung sa ating basidiospores. And our zygospores, these are rough-walled spores produced by the fusion of two identical cells arising from, take note, the same hypha. Okay? And the oospores, These are formed by heterogeneous fertilization. So here you have the fusion of cells from separate, non-identical hyphae. That is for our oospore. So, in review, pag sabi ko bumubuo ako ng acronym ko, Sir Edwin always try to put up with acronyms para lang hindi makalimutan yung mga terms. Sabi ko, pwede si sexual spores is BOAZ, B-O-A-Z, kasi may word dapat na pumasok. So that alam nyo na yun, si Basidiospore, si oospore, si ascospore, and then you have your zygospores. Those are your sexual spores. But still, you should know how they are produced. Kasi sabi nga natin ngayon, oospores, dalawang non-identical hyphae. Kanina si zygospores, parehong hyphae, identical hyphae. We get into our asexual spores. So what are our asexual spores? Generally kasi they're divided sana into two. You have your sporangiospore and then you have your conidiospore. So that pag sinabi natin kasi sporangiospore, ito yung mga na-form by successive cleavages. Yun yung mag-cleave within a sac-like head called a sporangium. Si sporangium, attached siya to a stalk which we also call now your sporangioform. Makikita nyo later on dyan sa ating slides. Tapos yung spores na initially enclosed dun sa ating sporangium ay marerelease siya when parang din dun sa Cleistotocium magra-rupture si sporangium. Okay? So si conidiospores kasi, when we're talking of the conidia, which you will again see in in the next slides, yung mga conidiospores, ito yung mga free spores, they are not enclosed by a spore-bearing sac. Okay, wala silang covering. So, nagde-develop sila either by pinching off the tip of a special fertile hypha or by the segmentation of a pre-existing vegetative hyphae. Examples natin ng conidiospores, you have there your arthrospore, you have your chlamydospore, and your blastospore. Okay, so ito na sila. You have here your arthroconidia. Ano ang importante kay arthroconidia? Dinrawing niyo din ito so that you have, if you notice, dun sa drawing ninyo, may empty spaces that appear between each arthrospore. This is what we call our disjunctor cells. So where do you see this arthroconidia or your arthrospores? They are with Coccidioides immitis and our Geotrichum candidum. Now, ito yung ating illustrations or, pictures of our arthroconidia. Okay? So, pag nag-break yan, yung sinasabi natin kanina, the most simplest form of reproduction, each of these, hiwa-hiwalay na yan, mabubuo ulit yan into another buong fungi. Okay, so blastoconidia. These are conidial formation through a budding process. So, daughter cells buds off the mother cell and is pinched off. Diba sabi natin ganyan, pinching off, yun yung mga mode of reproduction din. So, dito kasi you have the mother cell. Pag ini-imagine yung mother cell, meron munang uumbok. Diba? Before it gets to separate itself from the mother cell, kailangan munang merong multiplication ba tawag nyo doon, yung sa ating nuclei para ma-provide mo ng sariling nuclei si daughter cell. Okay? So, habang nagkakaroon ng division si nuclei para maprodusan ng nuclei si daughter cell, nagsishare pa yan sila ng cytoplasm, ng lahat ng meron si mother cell. Hanggang mabigyan si daughter cell ni nuclei, and then eventually, pag kompleto na siya on her own, pwede na siyang mag-pinch off. Now, another thing that you have to remember pag ganitong body is titignan nyo yung sa base kung saan sila nag-connect ni mother. Kasi merong narrow-based, ganyan, o kaya broad-based sa mga budding. Yung pag ganyan may mother cell at saka daughter cell. Kung saan nyo madidistinguish din yung iba-ibang fungi. Now, this, di ba sabi niya, daughter cell buds off the mother cell and is pinched off. Yan na naman nahiwalay na conidia. Mag-elongate na naman yan to form. It can elongate to form a pseudo hyphae. This is a characteristic of our Candida species. Then you have the Chlamydoconidia. These are large, thick-walled, usually spherical conidia produced by rounding up and enlargement of the terminal hyphal cells. Now, these Chlamydoconidia, they appear to be resistant resting spores. Ito yung sinasabi natin na parang nandyan sila sa state ba tawag doon? Na yan, hanggang pag unfavorable ang environment. Okay? So that pag okay na yung environment nila, then pwede ulit silang mag-germinate. So, we have a terminal Chlamydoconidia, also a sessile or sessile, however you want to pronounce it. And then we also have the intercalary. So, terminal, obviously, nandun siya sa tip ng hyphae, sessile, nasa tabi, si intercalary, nasa gitna or in between. Okay. And the sporangiospores, sabi nga natin kanina, di ba? They are formed by successive cleavages. So this is, you have there the mitotic spores produced within an enclosed, yun yung kanilang sac, sporangium, often supported by one, yung kanyang stalk or the stem. Tawag natin dyan, yung sporangiophore. And if you look at the picture, we have an aseptate hyphae. So wala siyang cross wall. So example, we have here our zygomycetes or now classified as mucorales. So yun yung ating mga ano na tawag natin? Asexual spores. Then, okay yan ang picture niya ng ating Rhizopus species showing sporangium on a long sporangiophore arising from a posiseptate hyphae. Okay? So, it says here, you note the characteristic rhizoids. Rhizoids yung parang roots kasi they grow, para din ito yung ating vegetative hyphae. Okay, so conidia. So, conidia, what are conidia? Or sabi ko nga, if you remember earlier, sabi ko conidia or spore. Sometimes they're used interchangeably. But of course, when we say conidia, yan yung ating asexual spores, or they are also called as our mytospore. They're produced either singly or multiply in long chains or clusters by specialized vegetative hyphae, which we call our conidiophores. Okay, and these conidia, they may be produced either from the transformation of a vegetative yeast or hyphal cell, or from a specialized conidogenous cell. What is a conidogenous cell? What is a conidogenous cell? Is a stella around? May myxostella. Pag sinabi natin conidogenous cell, wala si Stella. Asan si Jmar? Jmar? Wala din si Jmar. Nolan. Nandiyan kanina si Nolan. Maaga si Nolan. Nolan, what is a conidogenous cell?
Ma'am, in layman's term po, it is a cell that forms conidia, ma'am. Tama
Okay,
po ba,
or
ma'am?
produces. Yes,
Yes,
it's
ma'am.
any cell that produces
Produces
or
conidia.
becomes a conidia. Okay,
Yes,
thank you, Nolan.
You're welcome, ma'am.
Okay, so now this, with regards to the conidogenous cell, we have now your phyllid or your annelid. Now, dependent na naman ito. Madali mo lang sabihin niya na siya ay phyllid or siya ay annelid. Because when you see a phyllid, this is a flash shape. Take note of the word secondary segment. So, nakikita mo mamaya, kunyari yung mga, di ba yung mga, if you have a flower in your hand with the stem, tapos may, alam niyo, may sepal, ba yung tawag doon, yung nasa, mag-a-attach doon sa stalk. So, parang ganun din lang yun sa mga, kasi sabi nga natin, parang plant itong ating mga fungi. So, ito, pag nakakita kayo ng flash shape secondary segment, which produces the conidia, or from where the conidia will arise now. That would be already your Phylloconidia. Si Phylloconidia, kasi nga nag-arise siya from a phyllid. When you say an Anneloconidia, it arises again from an annelid. Yun yung secondary structure kung saan manggagaling o yung magpo-produce ng ating conidia or ng ating conidium. No. So when we say conidiogenesis, this is again, the word genesis in itself, it says the formation, it's a process. So it's a conidia formation, and it can be blastic or thallic. So that when we say it's blastic or thallic, Rangel, gisingin ko kayo para at least gising tayong lahat. Asan si Rangel? Wala si Rangel? Ralph, are you there? Ralph.
Good afternoon po, ma'am.
Yes, katulog ka na yata, Ralph.
Ma'am, hindi, ma'am.
Yeah?
Hindi po, ma'am.
O sige, blastic over thallic. In your own words. Biglang ganun, in your own words. Blastic conidiogenesis.
Ma'am, kapag...
Sige, go.
Ma'am, magbe-base na lang po ako sa term na plastic, ma'am. Kapag plastic, ma'am, parang yung mag-enlarge lang mismo yung parent niya, ma'am. Then yung thallic, ma'am, magpo-form siya ng, ay mauuna yung septum niya, ma'am. Parang ganun, ma'am.
Kasi sabi niya septum forms first, di nauna siya. Sige, go, Ralph. And, so ito, Ralph. Ibigay ko sa iyo yung next image. O, ayan, o. So, you have there, nag-choppy ka.
Ay, ma'am, choppy po, ma'am.
Ayan, okay ka na.
Ay, sorry po, ma'am.
Okay. Andiyan na sa images mo, you have the blastic and the thallic. Diba thallic from the word thallus?
Yes, ma'am.
Okay. Ang game na. Okay na?
Okay na po, ma'am.
Okay. So the illustration, yes. Ralph, pa-unmute. Pa-mute. Unmute. Okay, thank you. So you can see from the illustrations here the difference between your blastic and your thallic. So that dun sa blastic nyo, nakikita nyo kaagad yung conidium, parang budding lang siya. So it comes from the parent cell. Whereas sa thallic, magkakaroon mo na siya ng parang septation din, yung tinatawag natin kanina. Cross wall is laid down before differentiation of the conidium begins. Okay, so that's between your thallic and blastic conidiogenesis. So going back again to your conidia, aside from them having this thallic, you have the blastic, the Phylloconidia, and the Anneloconidia. We also have the micro and the macro. So ito wala nang malilito kasi you have the micro in itself. Sinasabi natin lagi pag micro, these are small, unicellular with a round elliptical or piriform shape. Pwede silang sessile din or pwedeng pedunculate microconidia. Sessile, parang kanina din, nag-a-arise siya sa tabi mismo ng hyphae, pedunculate, nasa dulo ng ating hyphae. Okay? Macro, dahil macro siya, malalaki, large, usually multiseptate, and club, oval, or spindle shape. Karamihan ng ating macroconidia ay spindle shape. There are thick or thin-walled, pwede rin silang spiny, which is appropriately termed as echinolate or smooth surface. So that's for our micro and macroconidia. Then, examples lang nito ulit. So you can see here now, again, you meet mentagrophyte kanina. Ang tinitignan ninyo ay yung kanyang hyphae. Ano ang hyphae ni mentagrophyte? Nides. Ano ang hyphae ni mentagrophyte as to shape? Si Robert. Asan si Robert? Job. Shane. Nag-roll call si ma'am. Shane, are you there? Vian. Wala din si Vian. Nag-check-in lang yata kayo, tapos nag-check out na ulit kayo. Rona. Naku, andyan pa si ano. Si Rona? O Robert, andyan ka na.
Hello, ma'am.
Oo. Nakatulog ka. Nakaidlip ka.
Ma'am,
Siguro nakahiga ka. Nakatulog ka na. Okay, Robert. Ano yung earlier na mention natin about the hyphae of fermentagrophyte?
Spiral, ma'am.
Okay, spiral. So here, ano naman yung nakikita natin sa picture niya ngayon? Yan yung kanyang? Anong present ngayon kay mentagrophyte? Although we do not see now the spiral hyphae. What are we seeing in the picture? Robert, yan yung kanyang?
Hindi ko alam.
Sabi ko na, natulog ka eh. Yan yung kanyang conidia. So that you have there a macro and a microconidia. So parang doon sa mga iba na rin natin nandyan, si Trichophyton rubrum at saka si Tonsurans. Yan kasi ang magdi-differentiate later on itong ating mga dermatophytes as to the presence or the absence or kung sino mas marami o mas konti. Parang sasabihin na rare ang micro kay menta or kay rubrum, mas marami siyang macro. Yung mga ganun. Yun ang kanilang part of their identification. So earlier, you have already mentioned about the Phylloconidia. Sabi nga natin, it comes from a phyllid, which is your conidiogenous cell. And also you have your examples lang yan ng ating Phylloconidia. And again, of course, your annelid, that is your conidiogenous cell. Conidiogenous cell. So ang kanyang conidia would be your anneloconidia. Okay. So after the classification of fungi, start sending your papers, activity 1 and 2 sa aking either sa aking, you can PM me privately, picturean nyo, send nyo na, and then, or you can send it sa group chat. Makikita ko naman yun. You had activity 1 and you had activity 2. Okay? So, classification, sabi ko nga sa inyo, that was your third question. You have botanical taxonomy, and of course, you have the type of mycosis. As to botanical taxonomy, you have Zygomycota, or now we have the Glomeromycota, or the Mycorrhizales, Ascomycota, Basidiomycota, and our Deuteromycota. As to type of mycosis, we have the superficial and cutaneous mycosis, subcutaneous mycosis, systemic, and opportunistic. For the Zygomycota, if you read onto your Mayhon, so medyo merong changes, but this one includes Glomeromycota, then and subphylum, Mucoromycota, and Entomotoracortina. This group of fungi, they produce the hyaline, sparsely septate hyphae. Now, it can undergo both asexual and sexual, so that pag asexual reproduction, ang kanyang spore would be your sporangiospore. Asexual, okay, thank you Ria, thank you Genevieve. Sexual reproduction, that would be your zygospore. Clinically important genera under Zygomycota, we have Rhizopus, Mucor, Rhizomucor, Rictimia, which was formerly under, thank you Azarina, Absidia, and lagi kong favorite si Kuning Hamela kasi ang sarap niyang sabihin. Okay, Ascomycota, these are ubiquitous in nature and produce true septate hyphae. They all exhibit a sexual form. Yung tinatawag natin teleomorph. They have a teleomorph, pero meron din siyang anamorph. So, asexual reproduction, conidia. Tandaan nyo na yung mga yan. Baka yan ang susunod na post-test ninyo. I'm developing different H5P. Thank you, Jmar. Sexual reproduction, Ascospores. So, nabulol na ako. Napakita na natin dito yung ating anamorph. Di ba? You have your anamorph, you have your teleomorph, you have your synanamorph. Going through a journal kanina, meron pang ibang morph. Hindi ko lang sabi ko, akala ko 3 lang. Teleomorph, anamorph, synanamorph. Meron pang isa. Thank you, Tam. Okay? So, for your Basidium mycota, they reproduce sexually through the formation of your Zygospores. So, meron generally a plant pathogen or environmental organism that rarely cause disease in humans. Nasa Deuteromycota kasi karamihan ng mga clinically important fungi. Thank you, Giselle. Clump connections occur at the septations in the vegetative hyphae. So, we have here examples, yung mga ating mushrooms, ras, smas, and kasama ang Ipropopocus neoformans complex. And Deuteromycota, aka, yes, thank you for all your photos, aka fungi imperfecti. It is not a true phylogenetic group, but rather an artificial class, lack asexual, sexual reproductive cycle characterized by their asexual reproductive structure, which is what? Their conidia. And they mostly resemble the Ascomyces. Example, you have your epidermophyton and our Sporotrix. Okay, the succeeding slides, you just have here the comparison of the fungal phyla important to human diseases. So, pinapakita dyan yung sexual structure nila. The spore, both asexual and sexual, whether meron siyang cross-wall o wala, so that malaman nyo kung siya ay septate or aseptate or sparsely septate. And then, ano yung mga representative genera. So, Ascomycota, growth characteristic is dimorphic. Ano ang kanyang asexual spore type? Conidia. Human pathogens, we have here Aspergillus, Blastomyces, and our Histoplasma. They're ubiquitous si Aspergillus, Blastomyces, hindi pa alam ano, Histoplasma sa soil, and so on and so forth. Malalaman nyo din naman yan sa mga succeeding lessons natin. Okay? So as to their clinical classification, we said again, as to the type of infection, so we were able to come up with the definition of mycosis as a disease caused by fungi categorized based on the site of infection. Kaya meron tayo superficial or cutaneous mycosis, which will be those that involve the hair, skin, or nails. Why? Because ito lang yung mga nag-infect ng ating mga keratinized tissues. Kaya doon sa ating KOH, when you do your KOH for your superficial fungi, yun ang role ng KOH. Okay, there is no direct invasion of the deeper tissues. Examples are those that belong to the superficial or cutaneous mycosis. We have dermatophytes, we have the tinea nigra, tinea versicolor, and piedra. Subcutaneous, they're confined to the subcutaneous tissue. Involve, thanks Joshua, involve the deeper skin layers including muscle, connective tissue, and bone. No dissemination to distant size. Dito papasok sa ating sporotrichosis, si mycetoma, si chromoblastomycosis, and our phaeohypomycosis. And systemic mycosis, as already stated, meron na tayo dissemination, and it can involve any organ. That the primary organ that is usually involved is the lungs. Okay? Why? Through the inhalation of the spore. And the infections caused by these systemic fungi would be your blastomyces, coccidioides, pinagsama ko si blastomyces and si coccidioides, sorry. Histoplasma, then you have your paracoccidioides and Penicillium marnifae. And the last but not the least, of course, we have our opportunistic mycosis. Infections occur primarily in patients with some type of compromise of the immune system. And since they're opportunistic, they attack the immunocompromised individuals. Nandito sa ating aspergillosis, candidiasis, cryptococcosis, and also your zygomycosis. So that ends my lecture. Another tabulation lang ng ating classification. So you have the superficial infection. Nahulog si cellphone. So the type of infection there is your pityriasis versicolor. Ang causative agent mo would be your Malassezia species. Again, and so on and so forth. Okay? Okay. So next topic will be on the laboratory identification of fungi. This is, bakit nagising bigla si Ces kanina? Wala siya nung tinatawag ko. Tapos ngayon nakakapag-send na siya ng photo. Ces, saan ka nang galing? Nawala ka kanina. Okay? So we have laboratory identification of fungi as our next topic before we go on to the different groupings of our superficial or our dermatophytes. You can get to your Mayhon, or I will be uploading. I started encoding the module last night. Nasa last part na ako. Hindi na kaya ng aking mata. Magti 3 o'clock a.m. na. So, sumurrender ang senior citizen. Natulog na muna. Sabi ni Ces, maingay daw sa room. Parang wala namang maingay. Good afternoon, ma'am. Thank you po. Thank you, C. So, read on the laboratory identification for those who have your book na kay Mayhon, or you can use your Baileys. Andyan din naman yan. And then, or you can read through the module that will upload. I will try my very, very best to finish it today. May lecture ako after your class, and then we have a meeting with Jed at 6 p.m.